In the previous lecture, we talked about the heterotrophic pathways of metabolism, and today we're going to wrap up that discussion of metabolism by talking about autotrophy. Just like we did last time, we want to relate the topics that we're going to talk about today to things that you learned about in general biology, and the autotrophy that you learn about in general biology is photosynthesis. In particular, you talk about photosynthesis as it pertains to plant cells, which undergo what we now know is oxygenic photosynthesis in their chloroplasts. And photosynthesis is talked in general biology as a combination of two pathways, one which is referred to as the light reactions of photosynthesis, where light and water go into the plant cell and oxygen is released, and the electrons and ATP generated in the light-dependent reactions move into the dark reactions or the light-independent reactions, where carbon dioxide is converted to a sugar using the ATP and the electrons generated in the light-dependent reactions. Those are all true, and we're going to build on those today, but of course we're going to take a look at things from a microbial perspective. So we're not going to be talking about plants, we're not going to be talking about chloroplasts, we are going to be talking about these pathways as they pertain to prokaryotes, and we're going to build on autotrophy. We're going to talk not only about photosynthesis, but about something called chemosynthesis, where chemical energy rather than light energy is used to convert CO2 to sugars, or to reduce that CO2 to sugars in what we call carbon fixation. So we're going to talk about photosynthesis as photoautotrophy, and then we will be also talking about something called chemoautotrophy. What all autotrophs have in common is their carbon and their electron sources and what they do with those electrons. So just like we could ask a couple of key questions of our heterotrophs in the last lecture, we can ask a couple of key questions here about the autotrophs. And question number one that we would ask them would be, what kind of carbon do you use? And all of them, regardless of whether they're using chemical energy or light energy, are going to be using inorganic carbon in the form of carbon dioxide. They are also going to be using an inorganic molecule as their electron source, but unlike the heterotrophs we talked about in the last lecture, these two molecules are not the same molecule. In our previous lecture, glucose as a stand-in for just an organic molecule was used as both the carbon source and also the electron source for heterotrophic metabolism. So the glucose was stripped of its electrons, and then the carbon skeleton could be used for all of the anabolic processes in the cell. Today, they're going to be using inorganic carbon, CO2, but they will be using a variety of different inorganic molecules as their electron sources. What they will, though, have in common is that they use those electrons and they use that CO2 to make food. They reduce the CO2 by grabbing the electrons from those inorganic molecules, and they make them into sugars, which of course they're going to use for their heterotrophic metabolism. So that idea of photosynthesis as energy plus CO2 plus water yielding a carbohydrate and water and oxygen still holds true. We just now understand that there's a lot more to it when we discuss these things from a prokaryotic standpoint. So what we're going to do is we're going to look at what all autotrophy has in common, and then we will look at different types of autotrophy. And what all autotrophy has in common is that all of these organisms make their own food from carbon dioxide. And so we refer to that in this lecture, and when we talk about metabolism, as CO2 reduction, to remind ourselves that the electrons that we're collecting from various places are being used to reduce that CO2. In the previous unit, we were talking about that phenomenon more on a global scale, and so we referred to that as carbon fixation. Either way, you're talking about the same thing. You're talking about converting CO2 into an organic compound, and there are three basic ways that that is done. The first way on the left is utilizing something called the Calvin-Benson cycle, and the Calvin-Benson cycle is the sole way that sugars are produced in plants and in plant-like protists and in some bacteria. Within the prokaryotic world, where we do have more metabolic diversity, we have an additional two ways that CO2 can be fixed, and they are the reverse citric acid cycle and the hydroxypropionate pathway. Regardless of the pathway that you're talking about a pathway that takes in CO2 and converts it to a sugar. Now, just as a point of clarification, the reverse citric acid cycle may sound a little strange to you because most people don't talk about the citric acid cycle. They call it the Krebs cycle. But the Krebs cycle is also known as the citric acid cycle, and if the citric acid cycle's job is to take organic carbon and oxidize it completely to CO2, then the reverse of that would be to take CO2 and reduce it to that organic molecule. So what all of these have in common is that they take CO2 and, utilizing a lot of electrons and a lot of ATP, they convert those CO2 molecules into organic molecules. So what they all have in common is that they need ATP. These are energetically very expensive processes, and that's the currency of the cell, which fuels the reduction of CO2, and of course the reduction requires electrons. So each of these has a hefty ATP requirement in order to make an organic carbon molecule. And we know from previous discussions that cells have only two ways of making ATP, either substrate level phosphorylation or oxidative phosphorylation and chemiosmosis. And of those two processes, only oxidative phosphorylation and chemiosmosis is going to provide for you the amount of ATP that is necessary to fuel carbon fixation, or the light independent portions of photosynthesis as it relates to your general biology discussions. So oxidative phosphorylation and chemiosmosis are the way that all of these autotrophs make ATP. So the heterotrophs use oxidative phosphorylation and chemiosmosis in order to make ATP, and so do the autotrophs. The differences between what we talked about in the last lecture and what we're talking about today is where the electrons come from and where they end up. In heterotrophy, the electrons come from food and they leave the system as waste. Today, we're going to be looking at a couple of different pathways where the electrons come from some inorganic source and are transferred to an electron carrier, which brings those electrons to the electron transport chain, and then those electrons are saved in order to fuel the reduction of CO2. So different source of electrons, different result of the electrons going through the electron transport chain. Now they're not garbage, we're going to save them. But once the electron gets into the electron transport chain, it flows through the electron transport chain, and that causes the translocation of protons across the membrane, which then flow through the ATP synthase, generating the ATP and the reducing power necessary in order to fuel carbon fixation. So if we want to talk about these autotrophs and understand more specifically what's going on, we can ask them where they get their energy. So we know they all make their own food, but where do they get the energy to do that? And they either get the energy to do that from chemicals, or they get that energy from light. So you might have a chemoautotroph, or you might have a photoautotroph. And where specifically do you get your electrons? Notice each of these is an inorganic molecule, either iron or sulfur or water. So you would have a chemolithoautotroph or a photolithoautotroph. They're all always using inorganic sources of electrons, so they're always litho. They're all utilizing an inorganic source of carbon, so they're all autotrophs. The difference between them is where they get their energy. If they get their energy from chemicals, they're called chemolithoautotrophs, and if they get their energy from light, they're called photolithoautotrophs. So what we're going to focus on is where they get the electrons and how they use those electrons, because that's really where the differences lie. And we're going to start with our chemolithoautotrophs, which should be new to you, and then we'll wrap up our discussion by talking about the photolithoautotrophs, which would include the plants and the plant-like protists that you discuss in general biology. So first up, the chemolithoautotrophs that use iron as an electron donor in order to undergo chemosynthesis. And one example of them would be Thiobacillus ferrooxidans, who oxidizes iron, as its name suggests, in order to put those electrons into the electron transport chain and go through this whole process that we've been talking about. So these guys have a protein on the surface of their cell membrane, which will pull in electrons from ferric chloride, and when they do that, it oxidizes the iron to ferrous chloride, turning it to the orange rust color that you see in this water in the inset picture. So this water is actually rust colored because of all of the iron that's being oxidized by the bacteria that are living there. And it just as a point of interest, notice that these guys live in an environment which has a pH of 2. So they live in an incredibly acidic environment. And just keep that in mind, because that's something that we're going to be talking about in future lectures, the ability of some organisms to not only survive but thrive in incredibly acidic environments. So for right now, though, he oxidizes iron. So his electron source is iron. That the electrons from iron flow through the electron transport chain, which causes the translocation of protons. The protons flow through the ATP synthase, which generates our ATP for us, and those electrons that come from iron don't become garbage. They are used to reduce NAD to NADH so that those electrons and the ATP that was generated can be shuttled over to the Calvin cycle in order to fix CO2. So iron oxidizers are one kind of chemolithoautotroph. The second kind of chemolithoautotroph is one who oxidizes sulfur, who should sound familiar to you because there are sulfur oxidizing bacteria that we talked about in the previous lecture unit who are capable of being the primary producers in deep sea hydrothermal vent communities, where they utilize the energy in the sulfur to drive the fixation of CO2. So one example of this would be Sulfolobus acidocaldarius, who also likes an acidic environment, and in addition to that, he likes it to be hot. His species name, acidocaldarius, tells you he likes it acidic, and he likes it like a cauldron. So he's a sulfur oxidizing bacterium that lives in acidic, hot environment. So he's what we would call a polyextremophile. He likes multiple extremes in his environment, which is something we're going to be talking about in our next lecture. So for right now, sulfur oxidation, which is a little more complex than the iron oxidation. Multiple different kinds of sulfur can be oxidized. So we'll just say sulfur is oxidized. The electrons go into the electron transport chain, which causes the translocation of protons across the membrane. The protons flow through the ATP synthase, generating ATP. The ATP and the electrons are saved, and both of those are used for the Calvin cycle. So notice that all chemosynthesis is done utilizing the Calvin-Benson cycle that is used by plants and protists and some bacteria that photosynthesize as well. Before we move on to photosynthesis, we want to just put this idea of chemosynthesis into the same schematic we were using in the previous lecture. So first of all, let's talk about that word chemosynthesis. Chemo refers to the conversion of chemical energy to ATP. So some chemical is used to make ATP. Synthesis refers to that energy being used to make carbon. So chemosynthesis, unlike photosynthesis, utilizes chemical energy to fix carbon. And the needs for that are ATP and NADH. ATP and a source of electrons that we would refer to as reducing power because they provide the electrons to reduce the CO2. And in the first example of chemosynthesis, we talked about iron oxidation, where iron, ferric iron is oxidized to ferrous iron, and you can see that it's been oxidized because it becomes more positively charged. Those electrons are donated to the electron transport chain, protons are moved across, ATP is generated. Electrons are not thrown away here, they are used to reduce NAD to NADH. The NADH and the ATP are used to fix carbon, and the fixed carbon moves over to the other portion of the cell where it can be used to fuel heterotrophy. Because remember, ATP can't be used as a long-term energy source, so you have to build up this sugar, and that serves as a longer-term energy source that can then be broken down into ATP later on when it's needed for the anabolic reactions of the cell. Likewise, chemosynthesis works very much the same with sulfur oxidation. Again, you have some inorganic molecule providing the electrons, the reducing power to the electron transport chain. Those electrons are used to translocate protons, which generates the ATP, and then those electrons are collected together in NADH molecules, and the electrons and the ATP go to the Calvin cycle to fuel carbon fixation, which then can be used to fuel heterotrophy and then the anabolic pathways of the cell. So that's chemosynthesis, iron oxidation and sulfur oxidation. Now we're going to move on to photosynthesis, which should be a little bit more familiar to you from general biology. So photosynthesis involves the production of ATP from ADP using solar energy. So a lot of times you'll hear it referred to as photophosphorylation. Phosphorylation, of course, is just a catch-all term for the addition of a phosphate group to a molecule. A DP adenosine diphosphate has two phosphate molecules, and it is phosphorylated to ATP, adenosine triphosphate. So there's the phosphorylation, and what fuels that is light, so it's called photophosphorylation. And there are two kinds of photophosphorylation that exist on the planet. The first is one that you would have learned about in general biology, which is called non-cyclic photosynthesis. That occurs in plants, that occurs in protists, and that occurs in the cyanobacteria, and is governed by a pigment called chlorophyll. You're going to learn in just a few moments about a second kind of photophosphorylation, cyclic photophosphorylation, which occurs only in bacteria. So, photophosphorylation uses electron transport chains just like respiration, which means you need electrons. And this is where those electrons that the iron oxidizers or the sulfur oxidizers or the water oxidizers are going to be needing those electrons, right? They need those electrons because those electrons are what go into these systems. So they don't eat food, they don't eat organic things, they eat inorganic things. And plants and protists and some bacteria eat water. and that water is oxidized, and the electrons are transferred to a chlorophyll molecule. And the beautiful thing about a chlorophyll molecule is that it houses this electron, and then it does the magical work of converting a photon of light into enough excitation, because it absorbs that light and that causes a conformational change in the pigment such that that electron becomes super high energy and is shot out of the chlorophyll molecule. And in non-cyclic photosynthesis, the chlorophyll molecule shoots out the electron, it lands into a second photosystem, and light again hits it, excites it even further, and then it gets shot so high that it can fall through the electron transport chain. So imagine, right, the only way that going through the electron transport chain is a passive process is if the electron is above the electron transport chain, and it's light that gets the electron above the electron transport chain. It's kind of like those games that you'll see at carnivals and fairs where you have a metal ball at the bottom of a really tall tower, and there's a bell at the top of the tower, and there's a lever, and someone will come by with a hammer and they'll slam the hammer down on the lever, and that will make that metal ball go high up into the machine and ring the bell at the top. And then of course that metal ball, because of gravity, will fall right back down to its place. So think of the metal ball as the electron, and think of the person swinging the hammer as the light. So the light comes and slams down on the chlorophyll molecule, and that shoots the electron up so high that it can fall through the electron transport chain. And that right there is the magic of photosynthesis. That's the conversion of light energy into chemical energy, because now you've converted the light energy into this potential energy in the electron, and then as the electron flows through the electron transport chain, the same thing happens that always happens. The electrons flow through, the protons are translocated across, the protons move through the ATP synthase, generating ATP, and instead of being thrown away, the electrons are used to reduce NADPH, NADP to NADPH. So here you have your ATP and you have your reducing power, and that is the light dependent portion of photosynthesis. And we know that that ATP and those NADPH are going to go to the Calvin-Benson cycle and be used to fix CO2. So that's plants and protists and bacteria. So bacteria exist there as well. Now, the other side of the slide, we have cyclic photosynthesis, which is a completely prokaryotic phenomenon. The beginning of this process is very similar to non-cyclic photophosphorylation. You have some inorganic molecule serving as an electron donor. So in this case, we have hydrogen sulfide donating an electron to bacterial chlorophyll, and light hitting the bacterial chlorophyll, right, and causing that conformational change that shoots the electron up such that it can fall through the electron transport chain. But notice here, instead of leaving right away, the electron cycles. So it cycles back down to the bacterial chlorophyll, gets excited again, shoots up to a high energy state, flows through the electron transport chain, goes back into the bacterial chlorophyll, gets excited again, gets shot up, flows through the electron transport chain. And so the same electron will cycle through this system multiple times, generating ATP. And then when there's a little bit of an ATP stockpile, the electron will move out of the electron transport chain and be used to reduce NAD to NADH. So notice in non-cyclic photosynthesis, water is oxidized, the electron moves through the system one time, does not cycle, and leaves as part of the NADPH molecule. In cyclic photosynthesis, hydrogen sulfide is oxidized, the electron flows through multiple times, generating ATP, and only then leaves the system as part of NADH. So one electron per ATP and NADPH molecule on the one side, one electron per many ATP molecules before it's ultimately used for reducing power on the other side. But either way, ATP is generated, reducing power is generated, and that ATP and that reducing power moves over to either the Calvin-Benson cycle or the reverse citric acid cycle or the hydroxypropionate pathway in order to fix CO2. Again, to relate what we've been talking about, respiration, fermentation, chemosynthesis, and now photosynthesis into that same schematic. So photosynthesis is the conversion of light energy to chemical energy, the conversion of light energy into ATP because of the work of that pigment that is able to absorb light and be changed by it and excite electrons through that change. Synthesis portion, same thing it is with chemosynthesis. It's the production organic carbon molecules from CO2. This process starts with inorganic electron donors, either hydrogen sulfide or water, which become oxidized. They donate their electrons to a chlorophyll molecule, right? And that's where the magic happens. That chlorophyll molecule excites the electron, allows it to flow down the electron transport chain, generating ATP. So there's the photo portion of it, collecting some electrons. And then those electrons and ATP are used for the synthesis portion, which, just like the chemosynthetic organisms, is used to fuel the heterotrophy of this photosynthetics. The photosynthesis that you talk about in general biology, we talked about at the very beginning of the last lecture, is now going to be called oxygenic photosynthesis, and that is because it utilizes water as an electron donor, which is oxidized to oxygen, so it generates oxygen as a byproduct of this metabolic pathway. The electrons are donated to chlorophyll, which gets excited and donates those electrons to the electron transport chain, right? Electrons are kept, ATP is generated, carbon fixation occurs, and that fuels heterotrophy. And in addition to that happening in the plant world, which we don't talk about in general biology, we said that this is possible in both the protists and some of the bacteria. On the left-hand side of your screen, you see a picture of cyanobacteria. Cyanobacteria are capable of oxidizing water in order to fuel their photosynthesis, even though they're prokaryotes, and so they can be oxygen-producing photosynthesizers. Now, their name might sound familiar to you because we talked about cyanobacteria in the previous unit when we talked about nitrogen fixation. We said that cyanobacteria might be members of a symbiotic relationship called a lichen, where they're fed food by a fungal partner, and that allows them to fix nitrogen. They can also, because they photosynthesize, be free-living nitrogen fixers because they have this relatively inexpensive way of generating ATP. Then they can use a lot of that ATP toward nitrogen fixation. And in fact, you can see a specialized cell there amongst all of the other cells of cyanobacteria called a heterocyst, which is a cell where nitrogen fixation occurs. In addition, we have oxygenic photosynthesis within our plant-like protists, and that exists in macroscopic red algae and green algae and kelp and all of those things, but it also, if you remember from our last unit, happens in phytoplankton, all of those microscopic organisms who photosynthesize on the surface of the ocean. Some of those are bacteria and archaea, but a lot of them are unicellular algae, and I've just put a couple here. The one on the far right are diatoms, which are really beautiful unicellular brown algae that they produce glass exoskeletons, and that's what gives them that really pretty shape. The other picture here is actually a satellite image. So the first two are micrographs. This is a satellite image, and on that image you might recognize the country of Italy, that boot-shaped landmass, and next to that you see this hazy light blue white milky stuff in the water next to it. That milky stuff, which is as large as this country of Italy, is actually a bloom, a huge growth of a marine alga called a coccolithophore. And the reason it's white and not green is that coccolithophores produce a calcium carbonate outer skeleton, like a baking soda outer skeleton, so it's white. And when enough of that is in the water, it's actually visible from space. In addition to some bacteria being able to undergo oxygenic photosynthesis, we learned today that they are also capable of something called anoxygenic photosynthesis, because instead of using water as an electron donor, they use hydrogen sulfide. So when they pull the electrons off of hydrogen sulfide, obviously they're not going to create oxygen, they create sulfur, which is a waste product, just like oxygen actually in this, but because it's not oxygen, this is called anoxygenic photosynthesis. And we don't care about the sulfur, just like we really don't care about the oxygen, we care about the electrons, which are donated to bacterial chlorophyll, which becomes excited by the sun, enough so that it's able to donate those electrons through the electron transport chain, right, which creates ATP, and then of course the electrons are kept and they are used for carbon fixation, and then that is used to fuel heterotrophy. And of the anoxygenic phototrophs, we have two big groups. One are the purple bacteria and one are the green bacteria. So you can see here two pictures of water. One is green, and that's because of the presence of green bacteria who utilize sulfur, and the other is red because of the presence of these small red spirilla bacteria who have grown to such an abundance that in both cases the organisms have actually colored the water. So we have purple bacteria and we have green bacteria, all of which are capable of anoxygenic photosynthesis using sulfur as an electron donor. So there's quite a bit to keep straight when it comes to photosynthesis. So I put this table together for you as a way to compare them, looking at four things. What is your electron donor? Do you produce oxygen? What kind of environment do you live in? And how do you fix your CO2? Now, just as a point of reference, all of the chemosynthetic organisms utilize the Calvin-Benson cycle. There's a difference here in the way that photosynthetic organisms do that. So of the eukaryotes, which include algae and plants, all of them utilize water as an electron donor and produce oxygen. And because they produce oxygen, they live in an aerobic environment and they all use the Calvin-Benson cycle to fix their CO2. Of the prokaryotes, we have the cyanobacteria, the green bacteria, and the purple bacteria. The cyanobacteria can actually use water or sulfur. If they use water, they undergo oxygenic photosynthesis and their environment is aerobic. If they utilize sulfur, they do not produce oxygen and they live in an anaerobic environment. But either way, they still use the Calvin-Benson cycle in order to fix CO2. The green and purple bacteria all use sulfur compounds. It's kind of funny because we actually have two groups of each of those. We have the green sulfur and green non-sulfur bacteria, which is really a misnomer because they all use sulfur. It's just one tolerates higher levels of sulfur than the other. Because they use sulfur compounds, they are anoxygenic phototrophs. They live in anaerobic environments, and in fact, for most of them, their pigment production is inhibited by the presence of oxygen, and so they will not grow in an anaerobic environment at all. And they either use the reverse citric acid cycle, which is the reverse of Krebs, or the hydroxypropionate pathway. Then we have the purple bacteria, which again, there are purple sulfur and non-sulfur bacteria. Because they use sulfur, they do not produce oxygen. They live in an anaerobic environment. Again, their pigments are suppressed in aerobic environments, although interestingly, some of them can survive undergoing heterotrophy in those environments, which we'll see in just a moment. And all of them utilize the Calvin-Benson cycle in order to fix CO2. So we finally finished talking about metabolism. And one of the reasons we do that is to highlight a concept that we talked about in the last unit. And that concept is the incredible metabolic diversity of the prokaryotes. So this is a chart that encompasses all organisms on the planet. And as I've alluded to earlier, there are a couple of key questions that we can ask any heterotroph and any autotroph and separate everybody into two groups. So the first question that gets asked here is, what is your energy source? And every animal or plant or fungi or anything on the planet will either answer that question, my energy source is chemical or it's light. If it's chemical, we call you a chemotroph. And then we ask the question, what's your carbon source? And everybody that is a chemotroph will answer that question, either my carbon source is organic or it is CO2. If your carbon source is organic, you are a chemoheterotroph. And then we can ask the question of all of our chemoheterotrophs, what's the final electron acceptor of your heterotrophic metabolism? And that can be oxygen or not oxygen. If it's oxygen, you undergo aerobic respiration and you are all of the organisms in kingdom Animalia, most of the organisms in kingdom fungi, all of the protozoan, the animal-like protists, and some bacteria. So most all of the eukaryotes fit right there in that box. If your final electron acceptor is not O2, not oxygen, then we ask this one final question. Is it an organic compound or is it an inorganic compound? If it's an organic compound, then you live fermentatively, and that might be that you're a fermentative bacterium like the genus Streptococcus. They all lack the ability to make an electron transport chain, so they never utilize respiration. You could also be Giardia, right? Giardia is a protozoan who lacks mitochondria and grows fermentatively all the time. And that's not to say that those are not successful ways of living, right? Those organisms would not be here today if they couldn't utilize those pathways to generate enough ATP to survive. But the key is they're all microbes. If we go back to that question, is it not oxygen and is it an inorganic compound? Again, when you answer that question, yes, it's an inorganic compound, you are a prokaryote. So whereas we're putting almost all of the eukaryotes in one box, the prokaryotes are already in all three of those boxes. And then if we go back up to our carbon source question, what's your carbon source, chemotroph? And you say it's inorganic carbon, and we say, okay, you're a chemoautotroph. There are tons and tons and tons of different ways that microorganisms can grow chemoautotrophically. They're all microorganisms, right? So where's the metabolic diversity? In the microorganisms, right? Let's go over to the light side. So the energy source is light, and you're a phototroph. What's your carbon source? Organic compound. So you are this crazy weird thing we call a photoheterotroph. Only bacteria do this. Most of those that are phototrophs use CO2, right? They're inorganic carbon source, and so we call them photoautotrophs. So we ask the question, do you use water, right? Yes, then you're an oxygenic photosynthesizer, which is all the plants, all the plant-like protists, the algae, and some bacteria. If you don't use water, you're a prokaryote. So we have basically two boxes where kingdom fungi, kingdom animalia, kingdom protista, kingdom plantae live, and then we have domains archaea and bacteria inhabiting all of these yellow boxes. So the metabolic diversity lies with the prokaryotes. And one last thing to say about metabolism, generally speaking, is we've hardly even touched the surface. This diagram, which I know you can't read all of these little things, but there are a bunch of little chemicals with arrows, right? So these are all chemical reactions governed by enzymes, and we talked about this. The red box encloses the primary catabolic pathway of glycolysis, the Krebs cycle, and the electron transport chain, and the green box encloses the primary anabolic pathway of photosynthesis. And so in spending 2 lectures talking about this information, we haven't really scratched the surface of all of the different things that every organism can do. So